All right, everyone, welcome. The major topic of this lecture video is the cell cycle and mitosis. You may or may not have heard of the cell cycle before, so I'm going to build it from the ground up. The cell cycle is the life cycle of our cell. Just as we have various stages of life, the cell cycle has its own stages that the cell will go through as it grows. We're also going to cover the concept of mitosis in this presentation, which refers to the actual process of cell division. Mitosis can be separated into four subphases, namely prophase, metaphase, anaphase, and telophase. It is through the four stages of mitosis and another process known as cytokinesis that our cells are going to divide into two identical daughter cells. We're going to spend some time looking at microscope slides and learning to identify these phases of mitosis. But before we get into the cell cycle specifically, it's very important that we discuss what a cell is, what makes it up, and what are the function of those parts. So when we consider the levels of structural organization, the cellular level is the second level of structural organization in the human body. The first level was our chemical level that included atoms and molecules, as well as the interactions between them. And it is from these atoms and molecules that we form larger structures known as cells. Cells are considered the basic unit of life. It is at the cellular level of organization that we finally start to consider these entities as live beings. Now, in the human body alone, there are very many different types of cells. But all cells have a few common components that are consistent among all of the types of cells that we have. All types of cells will be unique in their own way and serve different purposes for the body overall, but there are some components that are integral to the cell structure in general. Now, on the next slide, we will discuss the major parts of a cell, but we first need to understand what an organelle is before we go any further. So organelles are the tiny parts of cells that serve different functions for the overall cell structure. These act like very tiny organs inside of the cell that carry out different jobs. There are many different types of organelles, and some cells will even have unique organelles that other cells don't have. And it is the individual organelles that our cells have that give them their unique abilities. We will also find another substance known as inclusion that can be found in many different types of cells. Inclusions are any substances that are stored inside of the cell membrane. These inclusions are non-living substances such as granules, stored substances such as glycogen, and pigments. So the first thing that we truly need to understand about the cell are the principal parts that make them up. Most of the cells in our body are going to have these three principal components. So we'll always start with the cell membrane. The cell membrane can be found containing all of the structures of the cell. It is the cell membrane that is the boundary of what is not the cell and what is considered part of the cell. There are many organelles and inclusions that are contained within the cell membrane, and every cell is going to have one of these. We cannot have a cell without the cell membrane. It is the cell membrane that contains all of our structures and makes the cell its own individual unit. We'll also find the nucleus. The nucleus is a very unique structure in our cell and can often be seen under the microscope as a very dark round circle. The nucleus is often described as our cell's control center, and that's because the nucleus contains the DNA, which is the genetic code for our cell. The DNA inside of the nucleus directs all of the other cellular activities, and this is often thought about as the brain of the cell. It is the nucleus that directs our cellular actions. Now, not every single cell in the human body will have a nucleus, but most of them in fact do. We will focus specifically on the nucleus as we look into the process of mitosis and see how the nuclear structure will change throughout that process. Our third principal component of cells is what's known as cytoplasm. Cytoplasm is made up of both organelles and a substance known as cytosol. Cytosol is the fluid that all of our organelles are contained within. As I mentioned, there are many different organelles, and all of them are going to serve different functions. But those organelles are contained within the cell membrane and have to be suspended in some type of substance, and that is the cytosol. We have lots of things taking place in the cytoplasm, and I like to describe the cytoplasm as everything that is between the cell membrane and the nucleus. Anything inside of the cell that is not the nucleus is considered cytoplasm. The cytoplasm can be seen here surrounding the nucleus. So this slide depicts a cell with various organelles inside of it. This is a nice depiction to show us what some of these cellular structures are going to look like. However, I want to be very clear, there is no exact cell in the body that looks exactly like this one. This is a depiction, and every cell is going to look a little bit different. Certain cells are going to need more of certain organelles. For example, the mitochondria is the major organelle that's going to produce energy for our cell. Cells that need lots and lots of energy, such as muscle cells, will naturally have many more of these mitochondria compared to cells that don't need to make as much energy. So I'm going to take a second to describe some of these organelles that can be found on this slide. First and foremost, I want to go over the nucleus. The nucleus is our cell's major control center, and it contains the DNA. It is the DNA which is going to direct all of the other cellular activities. This type of nucleic acid regulates every single thing inside of the cell, and even what type of cell we're going to have. It is in the genetic code that will truly determine what our cells are going to look like and what components they will have. The nucleus actually has its own membrane called the nuclear envelope, which is a double-layered membrane that wraps around the nucleus. Inside of this membrane, we'll find lots and lots of little nuclear pores that allow for certain substances to come in and out, mostly RNA. mRNA is going to travel out of the nucleus and be used for protein synthesis later on. We'll also find a nucleolus inside of the nucleus, which is primarily going to be responsible for synthesizing another cellular organelle known as the ribosome. It is the ribosome that specifically will aid in protein synthesis. Inside of the nucleus, I've mentioned that there's DNA. However, DNA can exist in several different forms. And truly, the most common useful form is known as chromatin. Chromatin is a decondensed form of DNA that's easily accessible by proteins and other factors that will facilitate DNA replication and other cellular activities. I will discuss more about chromatin and its several forms later on. The next clear structure that I want to mention to you all is the plasma membrane. The plasma membrane is considered an organ as it wraps around the cell and contains all of its structures. You can see that there's quite a few organelles. This is a very diverse place inside of the cell. But in here we see organelles and this other substance. This other substance that all of our organelles are suspended within is known as cytosol. It is the cytosol that is the jelly-like medium that fills up all of the extra space inside of the cell membrane. Now, as we make our way into the cytoplasm, remember, cytoplasm is made up of both the organelles as well as the cytosol. As we start to look at some of these other organelles, we'll see that there's quite a diverse array of other structures. The first one I'll mention is the rough endoplasmic reticulum that's going to connect to the nuclear envelope. It is this rough endoplasmic reticulum that will be important for modifying different proteins and glycoproteins. This has several other actions, but we just want to get an idea at the moment of what each of these organelles looks like and where they can be found. The rough endoplasmic reticulum is named rough specifically because there are these little dark dots that can be found all over it, making it appear as if it is, in fact, rough. Those little dots are called ribosomes. Ribosomes, as I mentioned, are the primary organelle that facilitate protein synthesis. Naturally, since the rough endoplasmic reticulum has these ribosomes on it, we know that the rough endoplasmic reticulum is going to have some roles that relate to protein synthesis. But it is not just on the rough endoplasmic reticulum that we find these ribosomes. We will also find ribosomes free-floating inside of the cytosol. You can see a few over here and some over here as well. Now, not all of the endoplasmic reticulum will have ribosomes on it. In fact, we also have a structure known as the smooth endoplasmic reticulum, and that can be shown over here. Smooth endoplasmic reticulum will not have any ribosomes on it, and it's more so specifically going to be in charge of aiding with lipid synthesis and also detoxifying poisons. The liver, which is our primary organ for detoxification, is going to have lots and lots of this smooth endoplasmic reticulum. One of the most important organelles that you will learn about in Anatomy and Physiology 1 is the mitochondrion. Mitochondria are commonly referred to as the powerhouse of the cell. That's a common nickname for the mitochondria that you very likely heard at some point in your careers. This is a rather vague statement, so I do want to elaborate on that a little bit. It is the mitochondria that's producing all of the energy inside of our cellular structure. Our cell's energy currency is a molecule known as ATP. ATP is the primary source of energy that's going to drive most of our cellular mechanisms. And ATP comes directly from these mitochondria. Another structure in our cell that's actually not labeled on this slide, but I still want to mention because it's very, very prevalent, are vesicles. Vesicles are little bubbles that can be found inside of the cell, and they contain different things. They can move around the cell to transport substances in and out, and also around the cell to different organelles. Vesicles are used by many different types of cells, and we will commonly see them being used to transport substances in and out. The Golgi apparatus is one organelle that uses vesicles very frequently. The Golgi apparatus specifically serves as the USPS of our cell. This is primarily in charge of packaging up different substances and sending them off to where they need to go. This can be sending these substances inside of the cell, or perhaps sending these substances to other places outside of the cell also. Now, the last organelle that I'm really going to mention on this slide is what's known as the lysosome. The lysosome is a specialized type of vesicle that contains digestive enzymes. The digestive enzymes inside of the lysosome can be used to break down bacterial cells and other parasitic organisms that are trying to invade our bodies. Lysosomes are very useful when we need to destroy something dangerous that is infecting us. So hopefully from this slide you get just a little idea of all of the different types of organelles that can be found inside of the cell and what their major functions are going to be. Now that we have a little bit of an idea of what a cell is, what parts are going to make it up, and how the cell works, we're going to move into the cell cycle. Now keep in mind, the cell cycle refers to the life cycle of our cell. And just as the human has stages of life, the cell will have stages of life as well. We can think back to the stages of human life, such as adolescence, young adulthood, and elderly age, and understand that the cell cycle is set up very similarly. There are individual stages in the life of our cell that are going to facilitate different activities. You can see from this picture that the cell cycle is a continuous process that will eventually end up right back where it started off at and provide us with new cells. I'm going to go ahead and start right here as we go through the cell cycle. The first major phase of the cell cycle is what's referred to as interphase. Interphase can be looked at as sort of the adolescent phase of our cell, when the cell is growing up. And in fact, there are subphases of interphase shown on this slide which really suggest that. There are three subphases within interphase: G1, S, and G2. Specific events occur in each of these subphases of interphase, but the most notable of all of these is the S phase, which stands for DNA synthesis. It is in this S phase of interphase that we will actually be replicating our DNA, making a copy of our genetic code, and getting our cell prepared to separate those two sets of DNA into two daughter cells. We're going to see that through the process of mitosis, we take one cell and form two cells. And each new daughter cell is going to need its own set of DNA. And that's why we undergo this S phase. Now we also will have G1 and G2, which are both referring to growth phases. Different types of growth happen in each growth phase, and I'll elaborate on those in a little while. But I first want to make sure that we understand each phase of the cell cycle before we go forward. So it is interphase that is containing these three other phases, G1, S, and G2 in that order. Now after the G2 phase, we enter what's known as the mitotic phase. And the mitotic phase contains two processes, namely mitosis and cytokinesis. So mitosis specifically is referring to the division of our cell's nucleus. That is how specific we can get when we say mitosis. That is nuclear division specifically. Cytokinesis refers to the actual division of our cytoplasm. So even though these two stages of the cell cycle occur very close together, and you can see that cytokinesis will actually begin before mitosis is even finished, both of them refer to cell division as a whole. But mitosis specifically focuses on the division of the nucleus, while cytokinesis more specifically refers on cytoplasmic division instead. The four phases of mitosis are prophase, metaphase, anaphase, and telophase. The mnemonic you want to remember is PMAT. PMAT is the progression of these stages in order, and you will need to know these in order, so it's best to go ahead and get started in learning these phases right away. Cytokinesis will follow up mitosis, and after cytokinesis has taken place, both our nucleus and our cytoplasm have completely divided. We're left with two other cells that will both start their new lives in interphase, specifically the G1 phase of interphase. So every time we go through mitosis, we take one cell and turn it into two cells. And then each of those two cells will go through their own cell cycle and produce two more cells each. This is the never-ending process known as the cell cycle. Now let's take a second to elaborate on what's truly going on in interphase. Interphase, being made up of the G1, S, and G2 phases, indicates that our cell is going through lots of changes and seemingly getting ready to divide. The G1 phase is a period of a lot of cell growth and also metabolic processes. The G1 phase is necessary because our new cells are going to be entering back into interphase, and they have to start growing because they've just lost half of their mass. One cell split right down the middle into two. And so we have these very young, immature cells that need to be able to grow so that they have all of the inner contents that they're going to need. Now from the G1 phase, we enter into the S phase, and this is an extremely important phase I want you to have a good grasp upon. The S phase indicates DNA replication. So it is in this phase specifically that we take our set of DNA, specifically 46 chromosomes, and replicate each one of those, so that when this new cell is ready to go through mitosis, it has a set of chromosomes that it can give to each daughter cell. DNA is the genetic code of our cells, and we have to make sure that each cell has a full set of chromosomes that can direct the cellular activities inside. Now, after DNA replication has taken place, we'll then enter the G2 phase, which of course is more growth and specifically the replication of organelles. We're going to be dividing organelles so that we can give half of those organelles to one cell and half of our organelles to the other cell. So as we get into the G2 phase, our cell is really getting ready to divide through the process of mitosis. We are preparing for the upcoming division event that's about to take place. So we have to make sure that we're ready with two sets of everything so that we can give one set to our first daughter cell and one set to our second daughter cell. These are the events that compose interphase. Now, DNA replication is a process that's going to take place in the S phase of interphase. In this S phase, we're taking our entire amount of DNA and making an identical copy so that we have two versions of DNA inside of the cell. This, as I just mentioned, is going to be necessary so that we can give one of these DNA sets to each of our new cells. DNA exists in these segments known as chromosomes. You may have heard of chromosomes before, but our entire genetic code is separated into 46 chromosomes. Each human has 46 chromosomes. 23 come from mom and 23 come from dad. And it is through this process of DNA replication that we take a single chromosome and end up replicating it. Through the S phase of interphase, we take one chromosome and synthesize two sister chromatids. Chromatids are a replicated form of the previously decondensed chromatin that we discussed a few slides back. So if you are anything like me, at this point you might be a little confused with this chromatin, chromosomes, and chromatids. All of those words sound very, very similar and can be the cause of a lot of confusion in the field of genetics. So we're going to go into this slide and I'm going to explain the difference between what chromatin, what chromatids, and what chromosomes are. Chromatin is the decondensed form of DNA. This is necessary during interphase so that our DNA is accessible by different proteins and factors that will be necessary to replicate it. So it is shortly after the end of cytokinesis and the entry into interphase that chromatin is going to start being formed. The only difference is this is decondensed, and I sort of imagine that this looks like a ball of yarn or a bowl of spaghetti. Chromatin is how our DNA is existing most of the time. But when our cell is getting ready to divide, we have to package it up so it's in a form that can be more easily moved around. Just in the same way that if you're going to move residences, you want to pack up everything nicely into suitcases so you can take as few trips as possible out to the car, chromatin is condensing into chromosomes that will be much more easily transported than this big, loose ball of chromatin. So during the S phase of interphase, we're actually going to be making a copy of our chromatin. We can imagine that we have this red strand of chromatin, and through the process of DNA synthesis, we actually are going to make an exact identical copy, and we're going to depict the copy in green. So through this condensation of chromatin into chromosomes, we end up with a structure that looks something like this. But we want to keep in mind that this structure is showing us a replicated chromosome, not a singular chromosome. And so for that reason, I'm going to depict each of these colors around the individual chromosome that we're referring to. This is showing us a replicated chromosome. Naturally, chromosomes exist in this singular form, but when they are replicated during the S phase of interphase, we end up with two copies of the same chromosome. Again, one is depicted in red and one is depicted in green, but they contain the exact same information. The red copy and the green copy are exactly identical. I've just used different colors to show you that they are two different copies from the chromatin. So a chromosome is one set of the DNA. Now, specifically, we also have this term called chromatids. So chromatids is a term that we will often use when we're talking about a replicated chromosome. Two chromosomes that are exactly identical are often referred to as two sister chromatids. So hopefully this slide clears up a little bit of confusion about the jargon that we use here in the field of genetics. So we're going to start making our way into identifying each of these phases under the microscope. And we'll go ahead and show you this slide to show you a histological section of a particular group of cells. Now, despite the species that we're looking at, what we need to understand from this is that all of these cells in here are undergoing mitosis. Some of these cells are in different phases at different times. We don't have all of these phases of all of our cells going on at exactly the same time. I want to just show you this picture to indicate that all of our cells are at different stages of the cell cycle at different times. This cell is in a different stage than this cell, and that's why this one looks a bit different than this one does. We're going to learn how to identify what stage a cell is in the cell cycle based on the way it looks under the microscope. And that's what we're going to see on this slide. So as we progress through the stages of the cell cycle, there will be visible differences in the structure of our cells. The DNA specifically is going to be changing form throughout the cell cycle, and we can identify the different changes that occur in different stages of the cell cycle to tell us what stages our cells that we're looking at are specifically in. So we'll go ahead and start here with prophase. Prophase is the first stage in mitosis, and during prophase, our cell is going through the initial steps that are required to divide. So naturally, during interphase, we're going to have this chromatin that is existing, very decondensed. But as we progress into prophase, we're going to start to form what are known as mitotic spindles. Mitotic spindles will travel towards both sides of our cells, and they're going to grab onto the chromosomes to facilitate their division. We also start to see that the nucleus in prophase is going to start to break down, and we have the condensation of our chromosomes. One thing that we want to keep in mind is that these cell cycle stages is a progression of events. Our cells don't look at this life cycle as prophase, metaphase, anaphase, and telophase. The cell views these events as more like checkpoints. Once A happens, B can take place, and once B happens, C can take place. So the stages of the cell cycle are a nice convention that we use to understand where the cell is at, but we also have to understand that each of these stages of the cell cycle do occur in a progression. So it is at early prophase that we will often see what looks like this. But as we get closer to this second stage of metaphase, that's going to start showing that the nuclear membrane is starting to break down, and our condensed sets of sister chromatids are getting ready to line up along the center of our cell. And that's exactly what we see down here in metaphase. Metaphase has all of the chromosomes of our cell lining up along the center, or the equator, or the metaphase plate. You can refer to this structure as any of those. But all of these chromosomes are going to be lining up here along the center of the cell. And by this point, these centrosomes that are releasing these mitotic spindles have moved to either pole of the cell and are projecting these mitotic spindles to reach out and bind to each of these chromosomes. Specifically, these mitotic spindles bind to the center point here, known as the centromere. Now, as we progress on to anaphase, this is the first phase that we really start to have true cell division. We're actually pulling our sister chromatids apart. We're going to separate them to make sure that each of our daughter cells has a full set of chromosomes. As this phase progresses, our chromosomes are going to get closer and closer to the centrosomes, and we're going to start to form a structure known as the cleavage furrow. The cleavage furrow, you can see here, indicates that we are looking at telophase, which is our final stage of mitosis. As we progress through telophase, we're going to start decondensing our chromosomes, and we're going to form that nuclear membrane right around each of those sets of newly decondensed chromatin. So these are a couple of the events that we're going to look for in each of these stages of the cell cycle. Interphase is our first phase, and also our longest phase of the cell cycle. Interphase is consisting of the G1, S, and G2 phases. So in interphase specifically, we have different events that are occurring that are surrounding cell growth and synthesis of DNA. So during interphase, our genetic material is in the chromatin form, and that's going to be decondensed and therefore very thin. So we don't really see it that well here in the nucleus. We're going to have a very clear, crisp nuclear membrane because at this time, the nuclear membrane has not started to deteriorate. So in my humble opinion, interphase is the most boring phase under the microscope. We'll start to see that as we enter into the mitotic phases, that our cells are going to start to look a little bit more exciting under the microscope. So our next phase is prophase, and prophase is really starting to show us the first signs of cell division. You can see that in the nucleus, we're starting to condense our chromosomes a bit more. That's why we have these darker spots. Nuclei and chromosomes tend to stain much darker under the microscope. So we're going to start the process of chromatin condensation during prophase. We will see the spindle fibers beginning to form as they're getting ready to move to different poles to pull our chromosomes to different parts of the cell. But the other thing that we see is the destruction of the nuclear membrane. This is going to start to degrade and break down so that it's not preventing our chromosomes from moving around in the cell and traveling to different sides of our cell. So specifically with this picture, you can see a few more darker spots found within the nucleus, and you also might see some openings in the nuclear membrane as well. And this will help us distinguish prophase from interphase. Now, metaphase is probably one of the most distinguishable phases of all. Metaphase is very, very clearly showing all of the chromosomes in our cell lining up along that metaphase plate. There will be only one line of this genetic material. Remember, genetic material stains very darkly under the microscope, so that is what you're looking for. You can even see these spindle fibers that are attaching over to our centromeres of each of our chromosomal pairs, and the centrioles that are found at either pole of the cell. So compared to prophase and interphase, metaphase looks much, much different on the microscope. We only have one line of very dark genetic information. And now, because we have that condensation of our chromosomes, that's going to leave us with this much darker color. Now our next phase is anaphase. And anaphase is the first phase of true cell division. we actually start to have our chromosomes being pulled apart from each other towards either sides of our cell. And that's why we're going to see two sets of chromosomes on either side. You'll still see the spindle fibers, but you're going to only have half of the DNA on this side and the other replicated half on this slide. Anaphase and metaphase are two of the most uniquely looking phases of mitosis, and they tend to be the more simple ones to identify. You want to focus specifically on the genetic information and how it's arranged when looking at these different slides. Now, telophase is the last and final phase of mitosis. Remember, it is mitosis that's referring to nuclear division specifically. We are only focusing on the nucleus. Telophase is clearly shown when we start to have the cleavage furrow being formed. This indentation on either side of the cell will actually show you that our two cells are pinching off right at the center, and that's going to allow them to separate and form two independent cells. During telophase, we're going to start to reform the nucleus around our genetic information, and we're going to start getting rid of those spindle fibers because mitosis already has progressed and we don't need them anymore. It is during telophase that cytokinesis is also going to be taking place, as that refers to the division of the cytoplasm specifically. So hopefully this slide is a bit helpful to you all when trying to identify these different phases of the cell cycle and what events are going on in each. One thing that I really want you to keep in mind is that the cell cycle is a progression of events. So certain things will look different from early prophase to late prophase. We also have pro-metaphase, which is going to be right between prophase and metaphase, and certain events are going to be happening around that time too. But we put the different phases around mitosis so that we can keep a general idea of what events are going on in the cell at different stages. Now, to summarize the process of mitosis and to go over why we have to understand mitosis in the first place, is that we're always damaging our cells. Certain cells in our body are very, very susceptible to traction and other traffic that is caused by external stimuli. We need to have a way that we can continue to grow our cells and get larger, and also to fix and repair damaged cells or cells that have been completely lost. Mitosis gives us the ability to take one cell and split it right down the middle into two identical daughter cells. And they're not only identical to each other, but they'll also be identical to the original parent cell that produced them. This is due to the DNA replication that we have. Because we are exactly replicating our DNA during that S phase of interphase, and we separate our chromosomes equally between our two daughter cells, each of those two daughter cells is going to be exactly identical to the parent cell that produced them. So it is the process of mitosis by which our cells go through to replicate themselves identically. All of the cells in our body, except for the gametes, will undergo mitosis. And I will talk about gametes, which are the sex cells, in a few. So every single cell in our body, with the exception of gametes, which are the sex cells, will undergo mitosis. Gametes undergo an independent process known as meiosis, which we won't talk so much about. We want to focus on mitosis, as that is how most of the cells in our body will end up replicating themselves, growing themselves, and repairing themselves. So that will conclude our lecture presentation on mitosis. If you have any questions about the information presented in this presentation, I encourage you to reach out to me through a message, and I'm happy to help you understand any discrepancies you have with this information. I am always here to help, so please keep that in mind, and I will see you all in the next lecture presentation.